Viruses simplified. In this video, we'll try to simplify the virus. We'll concentrate on structure, replication, and pathogenesis. What is a virus? It's a small unicellular organism, an intracellular organism containing either a DNA or RNA. They cannot be grown on artificial media and they have a complex mechanism of multiplication. They don't have a proper cellular organization and they lack enzymes necessary for multiplication. So that will be taken up from the host cell. Now, let us dive into the structure of the virus. The basic thing what is required is a genetic material. It might be either a DNA or RNA, which can be a single stranded or double stranded. It can be linear or circular, segmented or unsegmented genetic material. This genetic material is covered by a capsid, which is basically made up of protein subunits called capsomeres. They protect the DNA from the environmental forces. In the non-enveloped viruses, this starts with the attachment of this particular capsid to specific receptors on the host cell, and this capsid as such is a antigenic property. Most of the viruses have an envelope surrounding this capsid, and which is a lipoprotein component. Means when the virus buds out, they will take up the lipid from the host cell and come out with this particular envelope. On top of this envelope, there can be some projections called peplomers, which are basically they are the attachment points of the virus to the host cell. There are some exceptions. Most of the DNA viruses are double-stranded DNA except for parvovirus, which contains a single-stranded DNA. RNA viruses possess single-stranded RNA, whereas the rotavirus has a double-stranded RNA. The retrovirus has two copies of a single-stranded RNA. Now, most of the viruses are enveloped, whereas the non-enveloped DNA viruses are parvovirus, adenovirus, and the papovavirus. The non-enveloped RNA virus are the picornavirus, reovirus, calcivirus, hepatitis A and E. Next, we'll talk about the symmetry. This is basically how the genetic material and the capsid is arranged. There are two major types of symmetry. One is a icosahedral symmetry, something like you can imagine like a football covering the genetic material. It is made up of some triangular facets and it is quite a rigid structure. The next one is a helical symmetry, where the genetic material is in the center and the capsomeres are coiled around the nucleic acid. Some of them can also have a complex symmetry. Now, depending on the size varies from a very small, which is the parvovirus, 18 to 26 nanometers, to a biggest virus around 200 to 400 nanometers, which is the pox virus. Shape-wise, there can be multiple shapes. The most common one is the spherical shape. You can also have occasionally different shapes like the bullet shaped in rabies virus, filamentous in Ebola virus, brick shaped in your pox virus, space vehicle shaped in adenovirus, wheel shaped rotavirus, star shaped astrovirus, and rod shaped tobacco mosaic virus. Now, when you talk about viruses, we need to differentiate that from prions. So, prions is basically abnormal infectious protein molecules without a nucleic acid. And they are very much resistant to the physical and chemical agents. And there are a few diseases which are caused by prions, which we will deal at a later stage. Viruses, when you classify them, basically you can classify them as a DNA, RNA viruses. I have marked a few which are very important for undergraduate medical students. So, herpes virus family, you have the group of alpha, beta, and gamma herpes viruses, out of which the HSV1, HSV2, varicella zoster, Epstein-Barr virus, cytomegalovirus is very important. Then you have the next, which is your hepatinovirus, which is your hepatitis B virus. Then you have the parvovirus, Papuaviridae family, which is your human papilloma virus, again very important, especially in the female population. Then you have the poxviridae. Smallpox has been eradicated, but still we need to know because it's the basic structure of the virus, and the adenovirus, which is the human adenovirus. Apart from this, you have a few viruses which are very important for undergraduate education. You have the picornaviridae, which is your polio virus, and hepatitis A virus is very, very important, including the Coxsackie virus. Calciviridae family, which is your Norwalk and hepatitis E virus. Togaviridae, the main component is rubella virus. Then you have the Flaviviridae. The most important members are the dengue virus and hepatitis C virus. Yellow fever virus is very important, and it is currently it is not seen in India, but it's worth knowing this particular virus. Coronavirus, we all know the very famous SARS-CoV, you have the MERS-CoV and COVID-19. The rhabdovirus, which is the rabies virus. Filoviridae, important one is the Ebola virus. Paramyxoviridae, the most important members are the mumps virus, measles virus, Nipah virus, and the Hendra virus family. Orthomyxovirus, the influenza virus A to D. You have the Bunyaviridae, the hantavirus, the Arinaviridae, which is your Lassa fever and other. The Riovirus, which is the rotavirus, and Retroviridae, which is the human human immunodeficiency virus. Now we look at the viral replication. The main stages of viral replication are the attachment, penetration, uncoating, biosynthesis, assembly, maturation, and release. Now we'll see one by one. The whole process of viral replication as one picture. Then the first step is the attachment, which is the most important step, where the virus will attach to the particular cell via receptors. So the HIV can attach to the CD4 molecules using a GP120 receptor. The influenza virus will have a viral hemagglutinin receptors which can bind to the host cells. The next step is a penetration. Now this can happen in three varieties. The first one is a phagocytosis. In virology is called as a viropexis. That is basically the whole virus is taken inside the host cell for the next process. The other type is the membrane fusion, like for example in HIV, where the virus completely fuses with the host cell and slowly the genetic material is released inside. Third, you have the injection of nucleic acid like bacteriophages, where the bacteriophage cannot completely enter inside the cell. So you have the virus sitting on top of the host cell, and the genetic material is alone is sent inside. The next step is uncoating, where the host cells will have the lysosomal enzymes which split up the particular virus into different components. This is followed by a process of biosynthesis. So in biosynthesis, what happens? There's a synthesis of nucleic acid, capsid protein, enzymes required for the viral replication, and also those enzymes which stop the host cell metabolism and dictate the host cell to produce only viral components. Now, if it's a DNA virus, so obviously you have a DNA which forms the mRNAs. The mRNAs will give rise to early proteins. Then the replication of DNA will happen. Then the late proteins will be formed. Now all these together, they will form the next generation of viruses. The next comes the concept of RNA virus, where there is a positive sense and negative sense RNA. So if the genetic material and mRNA have the same polarity, it is called as a positive sense RNA. Otherwise, it's called as a negative sense RNA. Now the difference is positive sense RNA, because it is the mRNA and the RNA are the same polarity. Immediately there will be a translation to form early viral proteins. Then there's a transcription to form an intermediary negative sense RNA, which is followed by formation of late viral proteins and the positive sense RNA, leading to next generation of virions. The negative sense RNA, what happens? There's a transcription using viral RNA polymerase. So positive sense RNA is formed. So this will lead to translation with viral proteins, which will also serve as a mRNA. Then the next process will be a transcription using viral RNA polymerase to give rise to negative sense RNA. The whole thing is assembled and the next generation of viruses are formed. A double-stranded DNA, so the strands will act, sorry, the strands will act as a positive sense and negative sense RNA with the influence of viral RNA polymerase with the formation of viral proteins. Then the complementary strands are formed, the complementary strands are assembled, and finally the next generation of viruses are formed. Retroviruses have a beautiful mechanism of replication. So they have a single-stranded RNA, which has to be now converted into a DNA RNA hybrid because it has to fuse with the host cell and slowly remain at the latency for some period. So then slowly the whole process of stopping the host cell enzyme production is carried out, which is now dictates the formation of viral proteins, and finally they are assembled. All these reactions or all these steps are courtesy of the DNA polymerase activity of the reverse transcriptase enzyme, which this particular virus possesses. The next step is assembly. Once all the required components are formed, they are packaged as one part to form the next generation of viruses. The DNA viruses assembly and things, the replication process will take place in the nucleus, whereas the RNA viruses, it will take place in the cytoplasm. Then comes maturation and release. So here there are two types. One type is the host cell is completely lysed and the complete viruses are pushed out. The next form, which is a more efficient format, is where there is a budding through the host cell membrane. Slowly, as the entered virus come out, they'll take up the lipid part of the host cell and come out and spread into the environment to go and attach themselves to the next host cell. Now, there can be some modifications can be happening. The two main things is mutations and interactions between viral genes and their gene products. Obviously, when the viruses are multiplying at a very high rate, there can be mutations, and this will be only visibly readable when there is either there is a difference in the clinical features or the death of the virus. The same kind of mutations can also happen when they are exposed to some kind of chemical or physical agents or to ultraviolet radiations. The main types of mutants what we are bothered about in medicine is the temperature sensitive mutants, where these are growing at a lower temperature but are not able to grow at a core temperature. These are used for the preparation of live viral vaccines so that they don't become infective at a core temperature. The next thing is interaction between the viral genes, where there can be three types. There can be genetic recombination, reassortment, and viral interface. Let's understand one by one. When there's a genetic recombination, when there is a virus which are somewhat similar or the same family, infect the same cell, what will happen? These viruses will exchange the nucleic acids between them and a recombinant form is formed, and these will be generally stable and will be able to replicate in the next cycles. The next one is a reassortment, when there is a same virus of two different strains. For example, we'll take up the influenza virus. So when they are formed, when the two different types of the influenza virus, they attack at the same time, there will be a reassortment, and this is most probably one of the reasons how the pandemic strains are formed and spread in a human population. Next comes the viral interference, where two different viruses attacking the cell at the same time can have a different kind of an effect. Sometimes one virus can dominate and the other virus can be downplayed, or there can be an enhancement of effect because of one virus on the other. Now let's talk about the pathogenesis, how the particular infection enters into the body. Once it enters, it can cause a primary infection, which can spread to a secondary site and then finally manifest as disease. So if you look at the respiratory route, most common things are the ones which are causing infection at the site of entry will be all your influenza type of viruses, rhinovirus, adenovirus, COVID, HSV, etc. Now, the viruses which are entering via the respiratory route but cause infection elsewhere is your measles, mumps, rubella virus, varicella zoster, cytomegalovirus, and smallpox virus. Same way, oral route, you have the gastroenteritis, which is caused by rotavirus or adenovirus calcivirus astrovirus, which causes infection at the portal of entry itself. The other ones, that is, they can spread to a distant location and can cause infection, is your poliovirus, Coxsackie virus, hepatitis viruses, cytomegalovirus, and Epstein-Barr virus. Same way, you have the cutaneous infections, which is because of the herpes simplex virus, human papilloma virus, and molluscum contagious. Because of any kind of bite, the infection doesn't happen at the portal of entry but happens at a different area, which is your examples: your dengue virus, chikungunya, Japanese encephalitis, yellow fever, and Weisseneur forest disease. Sexual route, again, there can be lesions in the primary area in herpes simplex virus and human papilloma virus, whereas hepatitis B, C, and D and human immunodeficiency virus will cause lesions at a different place. It is your the bloodstream. The same way, you have the transfusion-related reactions because of hepatitis B, C, and HIV parvovirus, and transplacental route of transmission can cause infection because of rubella, cytomegalo, herpes virus, varicella zoster, etc. Okay, so there can be some viruses which are producing infection at the site of entry, and some of them which will travel to another part of the body and cause infection. So let us understand this concept. So first of all, when the virus enters inside, there can be a local spread in the area where it is entering the body. The virus from then on will slowly travel to the lymph nodes, which is draining that particular area, and from there it will enter into the bloodstream. So this is called as a primary viremia. So from the primary viremia, they will go into the organs of reticuloendothelial system and they will multiply there, and they will come into the bloodstream once again, which is called as a secondary viremia. From this stage, they will go to the target organs, like wherever they are bound to cause infection, they will go to the particular target organs and they can cause infections and basically come up with all the clinical symptoms. Same way, you have to also understand that there is a particular viruses have affinity for particular types of cells. Like for lymphocytes, the Epstein-Barr virus, cytomegalovirus, hepatitis B, JC, and BK virus have an affinity like that. Now, once the virus is entered inside, they have to multiply and go ahead and infect more and more cells. So that happens where there is a local infection. For example, influenza virus, which is shed in respiratory secretions. If it is a bloodstream, the viruses can spread from the site of infection, site of entry into the blood, and then slowly start multiplying and going in for a secondary viremia. Sometimes they can be near the target tissue or the organ, like varicella zoster, mumps, or cytomegalovirus. Sometimes they can be the virus life is a dead end with that particular infection, which is your example is rabies virus. Now let's look at the incubation period. So from the time period when the virus entered the body to the first clinical manifestation is the time interval, is the incubation period. So it can be short if the distance which the virus has to travel is very short, like influenza virus. It enters via the respiratory tract and the infection is immediately caused there itself. Sometimes it's a longer, say for example, rabies virus, where the virus has to, the bite of the animal is on the foot and the CNS has to be affected, whereas the distance it has to travel is long with 2 millimeter per hour speed. So that can be a longer incubation period. Sometimes there can be exceptions also, but majority is Some of them have a longer incubation period if it is from a distance. Now let us look at the clinical manifestations. Mainly it can be a inapparent infection, means a subclinical infection not giving rise to any kind of clinical symptoms. The second type is apparent infection, which can be acute, means the disease is immediately caused, or subacute, where the disease lies dormant for some time without much of clinical symptoms, or it can be chronic, where the disease can spread over a period of months. So clinically, respiratory viruses like your influenza and coronavirus can cause the lower respiratory tract and upper respiratory tract infections. Gastroenteritis can be caused by rotavirus and norovirus. Hemorrhagic fevers by dengue, Ebola, like that. And some viruses which are specific for nerves can cause meningitis like your enterovirus or encephalitis viruses. Now, the pathogenesis as such, the first and foremost thing can be an abortive infection where the virus is not really compatible with the host cell, so the infection does not proceed at all. The next one is the cell death, where it can be a lytic infection. So what happens here is the host cell DNA will be completely inhibited, and there will be formation of the cells will be destroyed, and the cells will nearby will clump together, forming a multinucleated giant cells, what we call as a syncytia. So this allows the virus from spreading from one cell to another cell. There can be a immune mediated lysis, where the virus infected cells are killed by the immune system, the host immune system. The next one is a steady state infection, where the host cell and virus somehow try to live in a harmony without much of a cellular injury, which can cause either clinical or subclinical infection. Then you have a persistent viral infections, where the latent infections, the virus can stay inside the cell, and on and off it can give some kind of a increase in virus output and clinical symptoms. There can be a transformation of cells, where the virus infected cells can indefinitely divide to form a tumor, or there can be a latency like in HIV infection, where it is integrated with the host cell chromosome, and after a long period of clinical latency it can give out symptoms. They can be also seen in slow virus infections, and there can be sometimes a persistent tolerant infection, where the host is tolerant to the virus and there is no immune response to that particular virus. The viruses can also bring about some changes in the host cells, which is called as a inclusion body formation, which is seen with the histopathological stainings. Now this is when the virus, the viral proteins and other products will clump together and give a particular staining property to the cell, and based on this we can also find out the actual virus which is causing infection. They can be intracytoplasmic inclusion bodies or intranuclear inclusion bodies. The very famous one is a Cowdery type A and type B inclusion bodies. These are some of the most commonly seen inclusion bodies. Very important one is the Negri bodies in rabies virus. You can also see Guarneri bodies in vaccinia virus, molluscum bodies in molluscum contagiosum. The poliovirus and adenovirus can give rise to Cowdery type B inclusion bodies. They can be owl-eye appearance in a cytomegalovirus and measles virus. Now this is about the first part of the virus simplified. I hope this video was short and understandable. Do like and subscribe to my channel and share this video among your friends and people who would be interested. Thank you.